We are first going to take a look at the process of diffusion. Diffusion is considered to be passive transport. That means that we will have the passive movement of molecules to spread out evenly across a membrane. No energy is going to be required for this, and we say that the molecules will be moving down a concentration gradient. That means that they will be going simply from a high concentration area to a low concentration. Many nonpolar molecules can freely diffuse across biological membranes. They can diffuse by what is termed passive or simple diffusion. Remember, nonpolar molecules can cross easily unhindered. They don't require any proteins to do so. So they simply go from an area of high to an area of low concentration all on their own until they reach an equilibrium. However, anything that is polar or anything that is charged cannot move across the membrane without the use of a protein. In these instances, you will need a channel in order for these molecules to pass. In this case, we call this facilitated diffusion. The movement of these molecules is going to be facilitated by a protein. This image here depicts the concept of diffusion. In the top of the image, we have the movement of one solute molecule. So notice that the molecule in orange moves from the left to the right, meaning that it moves from a high concentration to an area of low concentration. The molecules keep moving until an equilibrium is reached, or the solute concentration is equal on both sides of the membrane. Please note that that doesn't mean that these molecules stop moving at equilibrium. It simply means that the number of molecules moving to the left will equal the number of molecules moving to the right. If we take a look in the second diagram, we have the diffusion of two different molecules in two different directions. So these small nonpolar molecules, the orange one will still be moving from an area of high concentration to low concentration, meaning it is moving towards the right, whereas the purple molecule is moving in the opposite direction, but still from an area of high to low concentration. Once again, these molecules will diffuse across the membrane until an equilibrium is reached. This image here depicts the same concept, however, with a channel protein instead. So in this instance, we have extracellular fluid that consists of a solute. In this case, this solute would be polar or it would be charged. Because it is polar and charged, it cannot easily cross through the membrane without being facilitated. In this case, we have a channel protein that will allow for the movement of that molecule once again from an area of high concentration to an area of low concentration. We can also have the use of a carrier protein, and a carrier protein can open or close to allow for these molecules to pass through, as opposed to a channel protein that simply remains open. Once again, we have the movement of the molecules from an area of high to low concentration. Now we will discuss the concept of active transport. In active transport, we have the use of energy in order to move the molecules. This is because the molecules are moving up the concentration gradient or against their concentration gradient. So in these instances, you are going to have molecules going from an area of low concentration to an area of high concentration. This is in the opposite direction of passive transport, which typically goes from high to low concentration. So as a result, you are going to have the use of energy. This energy usually comes in the form of ATP. As a result, we are going to have pump proteins that will help pump ions or molecules from their area of low concentration to an area of high concentration. We are now going to take a look at the concept of osmosis and how this differs with respect to different gradients that you can have. Osmosis refers to the diffusion of water across a membrane. Molecules are always in random motion, and this will lead to the diffusion of molecules. If a molecule accumulates more on one side of the barrier or membrane, this will create what is termed a concentration gradient. This will be important for allowing for osmosis to occur. Osmosis acts to reduce this concentration gradient in order to achieve an equilibrium. Keep in mind that active transport will be very important in building up a concentration gradient. This slide here depicts the concept of osmosis. So in the top left, we have a sugar molecule that is dissolved in water. If you take a look, you can see a synthesized membrane that is at the bottom of that U-tube. On the left of the U-tube, we have the sugar molecule in a very low concentration. However, on the right, we have it in a much higher concentration. Please note that this membrane is selectively permeable. A sugar molecule is considered to be polar, so as a result, the sugar molecule would not be able to cross through the membrane. However, the water molecules would be able to. In this instance, what will occur is the solute won't be moving from one location to another. It is the water molecules that will be moving, and they will be moving in a direction to reach an equilibrium. As a result, the water molecules are going to be moving towards the area of higher concentration of solute to be able to dilute it. This leads to osmosis, or the movement of water molecules. Along with the concept of osmosis, within cells we have what is termed osmoregulation. Cells will be able to regulate their water balance, and this is to maintain optimal conditions. Within the concept of osmoregulation, it is important to know the difference between these three terms that can be used to describe a solution. So imagine that a cell is being placed in a beaker of solution. We can describe that solution in one of three ways. That solution in that beaker can be hypotonic, meaning that it has lower concentration of solute. It can be isotonic, meaning that it has an equal concentration of solute. Or it can be hypertonic, meaning that it has more solute. When the cell is placed in that beaker of whatever solution you have, water will either rush in or out of the cell, and it will do so by passing through the aquaporins. So a cell will try to maintain optimal conditions. How it will do so will be different depending on what cell type that you have. So for the purpose of this course, it's important to note what these solutions are, what they describe, and to also be able to answer any questions with respect to either an animal cell, such as a red blood cell, or a plant cell being placed in either of these solutions. You should also be able to describe what are the optimal environments for each cell type. This diagram goes through those concepts. So let's first take an animal cell or a red blood cell and take a look at what it would do in each of those three conditions. So imagine that we take an animal cell and we place it in a beaker that is full of one of the three different types of solutions. So let's consider first if we have it in an isotonic solution. If we had it in an isotonic solution, that would mean that the concentration of solute would be equal inside and outside of the cell. So let's say that we have 60% solute inside of the cell. That means that our solution would also have 60% solute. Let's also consider the opposite. If I have 60% solute in my cell, that means I have 40% water. Outside of my cell, if I also have 60% solute, that would also mean I have 40% water. So if I have 40% water inside of the cell, 40% water outside of the cell, water is going to already be in equilibrium. That means that the water molecules that are rushing out of the cell are going to equal the water molecules coming into the cell. As a result, the cell is going to maintain its shape and be in the normal cell condition. However, if I take that cell, and remember we have 60% solute and 40% water in our cell, if I were to place it in a hypotonic solution, that would mean that I would have less solute outside of the cell. So if I have 60% solute in my cell, that would mean maybe I have 40% solute outside of my cell. Remember, if I have 60% solute in my cell, I also have 40% water. If I'm placing it in a hypotonic solution, that would mean that an example would be I have 40% solute outside of my cell in solution, which would also mean I have 60% water. So if we take a look at the water content inside and outside of the cell, I have 40% water in the cell, 60% water outside of the cell. Water is going to go from an area of high to low. That means in this instance, water is going to rush into the cell. My cell is going to lyse, which means my cell is going to burst. So that is not a good situation to be in if I was a red blood cell. On the other hand, if I were to place that red blood cell in something that is hypertonic, that means that by definition, that solution would have more solute. So going back to my example, inside of the cell I have 60% solute, 40% water. If I place it in a hypertonic solution, that means that I would have maybe 70% solute outside of my cell and 30% water. So examining the water content to figure out where my water molecules are going to go, I have 30% water outside of the cell, whereas I have 40% water inside of the cell. Water is going to move from high to low. It's going to move from 40% to 30%. That means my water is going to be moving outside of my cell into the surrounding solution, so as a result, my cell is going to shrivel up. So if I was a red blood cell, my ideal condition would be to be placed in an isotonic solution. On the other hand, let's take a look at what a plant cell prefers. So using the same concentration idea, if I were a plant cell placed in an isotonic solution, I would once again have water moving in and out of the cell at the same rate. So though this would be an okay situation, I would not have my full shape attained as a plant cell. However, if I'm placed in a hypotonic solution, I would have water rushing into the cell. As a plant cell, this would be ideal. Why? Because I have a central vacuole that is present to store all of this water. And the more water I have, the more turgor pressure I have exerting on the cell wall, which means that the more pressure I have, the more that I can take on my optimum shape. So if I were a plant cell, I would want to be placed in a hypotonic solution. I would want to take in as much water as possible to maintain my optimal shape. On the other hand, if I were a plant cell placed in a hypertonic solution, that means water would be rushing out of the cell, my central vacuole would be very small, and I would eventually get to the state where my cell wall, my cell membrane would be retracting from the cell wall, and we would call that a plasmolyzed state. So if I were a plant cell, my optimal condition would be to be placed in a hypotonic solution. Let's take a look at transport proteins. Transport proteins can be used for passive or active transport. When they're used in passive transport, this is also called facilitated diffusion. Remember, this does not require any energy. Molecules are moving from an area of high concentration to an area of low concentration. We saw this with our images with a protein channel or a protein carrier. Some of these proteins can be very specific with the molecule that they're transporting, such as aquaporins. However, some of them can be non-specific, meaning that maybe they allow positively charged ions through. It doesn't matter what that ion is as long as it's positively charged. Some can also be gated, which means that they require some sort of stimulus to be able to open or to close. Transport proteins can also be used for active transport. Remember, this uses energy, usually in the form of ATP. And this is very important in building up a concentration gradient. We will see how important this becomes when we take a look at building up protons on one side of the membrane in photosynthesis and in respiration. Many of the proteins involved in active transport are going to be very specific, but many of them can transport one or two things or three things at a time. So we call this active transport or co-transport. A good example of this would be the sodium-potassium pump. This is a pump that uses energy to pump both sodium and potassium from an area of low concentration to an area of high concentration. Here we have our example of both facilitated diffusion and active transport. So in the image on the left, we've got facilitated diffusion using a carrier protein or a channel, once again moving from an area of high concentration to an area of low concentration and no energy is involved. Whereas the image on the right involves active transport, moving a substance from low to high concentration. Once again, this is building up a concentration gradient, and this would require the use of energy, here in the form of ATP. Co-transport is an important concept. This is where a pump can be used to move more than one molecule. This can either be done in the same or opposite direction. So if you have two molecules moving in the same direction, we refer to this as symport. If they are moving in the opposite direction, we refer to this as antiport. A good example of this is a sodium-potassium pump. Some of these pumps use ATP, and some of these pumps will use a gradient as energy in order to pump something else. This image here depicts the action of the sodium-potassium pump. Important things to keep in mind is what is the concentration of sodium and potassium both inside and out of the cell. Remember, this is active transport, which means that these molecules are going to always be moving from an area of low concentration to an area of high concentration. If we take a look at image number 1, this represents the resting state of the pump. The resting state of the pump is to have the conformation where it is open to the interior of the cell. In this conformation, it has a high affinity for sodium ions, which means that sodium ions are going to be attracted to the protein. 3 sodium molecules are going to bind from the inside of the cell. Once this occurs, ATP is going to provide energy for the conformation of that pump to change. It is going to then open to the exterior of the cell, and as a result, the sodium molecules are going to leave. When the protein is in this conformation, it has a high affinity for potassium ions. Potassium ions are going to bind to the protein from the exterior of the cell. The protein is then going to change conformation as that phosphate group leaves the protein. You can see in figure number 4, there is a P in the bottom of the protein. That stands for a phosphate group. So as that phosphate group leaves, the conformation of the protein is going to change back to its original state. As a result, it's going to open up to the interior of the cell, and those potassium ions are going to be brought in. So a good hint to remember what the concentration is. If we think of sodium, sodium is something that is readily available. We have lots of salt in our diet. So as a result, the salt content is always going to be high outside of the cell. However, potassium is a more limiting kind of element. And so potassium ions you're going to want to conserve it as a cell. So in general, if you're thinking of yourself as a cell, you're going to want to get rid of excess sodium and pump it to the outside of the cell. But when you encounter potassium, you're going to want to take in as much as possible. So that might help you remember in which direction that these molecules move. Sodium will want to get out of the cell and potassium will want to come in. Remember, by definition, this is active transport, and that means that we are always going from low to high. So keeping that in mind, that means that if I'm pumping sodium out and this is active transport, the concentration of sodium inside of the cell must always be low and the concentration outside of the cell must always be high. Vice versa, if I'm taking potassium in and this is by active transport, that means that I must have low outside of the cell and high inside of the cell. This slide here illustrates the concept of co-transport and also of pumps. So here we've got a proton pump, and proton pumps are important in creating a concentration gradient. So once again, we have active transport, we have the use of energy, and we are going from an area of low to high concentration. So that means the proton pump is going to be responsible for taking hydrogen ions or protons from the inside to the outside of the cell. This is going to help create a concentration gradient. A good example of how this becomes important is looking at the proton sucrose cotransporter. This cotransporter will take advantage of the concentration gradient that exists because of that proton pump. So things are going to want to go from an area of high to low passively. So as a result, these protons are going to want to flow back. Once that concentration gradient is created by the proton pump, those protons are going to want to go from high to low back into the cell. This cotransporter will take advantage of that transport and attach that to a sucrose molecule. Remember, a proton is positive, so that will not be able to cross the membrane without some sort of protein. Sucrose is also considered to be polar, so that won't be able to cross without a protein either. So this cotransporter will take advantage of that concentration gradient and allow for sucrose to enter into the cell as well. Gradients are very important, and they can be used for many different functions. When a gradient is formed, it can be used for energy, such as the proton pump. So having active transport and creating a gradient, we will see in the process of photosynthesis and respiration that this is very important in allowing for these processes to occur and for allowing for ATP production to occur. When we have a gradient of ions, we can also refer to that as an electrochemical gradient. Now we're going to take a look at the transport of large molecules. So when we consider, just a bit of a recap, nonpolar molecules can easily diffuse through the cell, and they're going to be diffusing through simple diffusion from an area of high to low concentration. If we have anything that is polar or charged and small, that will pass through the membrane using facilitated diffusion. We can also have active transport to go against the concentration gradient and use energy to pump things from low to high. However, all of those mechanisms are reserved for small molecules. Sometimes a cell would like to get large molecules into the cell, or a large volume of molecules into the cell, or specific molecules. In this instance, the cell will need to use endocytosis or exocytosis. So this is when the cell will need to form some sort of vacuole in order to move these molecules back and forth across the membrane. Exocytosis specifically refers to secreting large molecules. So these vesicles are going to fuse with the plasma membrane and release their contents to the exterior of the cell. Very important in getting rid of waste material. Endocytosis is the opposite of this process and involves forming vesicles from bringing things outside to inside of the cell. And as this crosses through the plasma membrane, a vesicle will form. Phagocytosis refers to cell eating. This is a form of endocytosis. As we can see here in the image, this amoeba forms a pseudopodia, or a false foot, that will then go and engulf the algal cell. Eventually, it will engulf it all the way around. The cell membrane will pinch off, and this algal cell will be brought into the cell. As it is brought in, the plasma membrane will surround it, so it will then be enclosed in a lipid bilayer or a vesicle. Small dissolved molecules can also be taken into the cell by what is called pinocytosis, or cell drinking. This is when the cell can bring in small droplets of dissolved substances. The cell can also bring in specific molecules using receptor-mediated endocytosis. This is where there are different specialized pit areas in the plasma membrane that have specialized receptors. Once these receptors bind to their specific molecule, a vesicle will form, bringing in these contents to the interior of the cell. This image here depicts the various types of endocytosis that you can have within the cell. Phagocytosis is the process of taking in a large molecule of food or other particle. As this molecule is ingested into the cell, it is going to have a plasma membrane portion of it enclosed around it, which would now be termed a food vacuole. Eventually, this vacuole is going to fuse with lysosomes that will help digest the material. Pinocytosis is where large volumes of dissolved substances can be taken into the cell, once again, like cell drinking. And if the cell wants to take in specific molecules, that's when receptor-mediated endocytosis will occur, using specific proteins that will attach to specific molecules. Once you have the molecule binding to the protein receptor, this will then invaginate to form a vesicle.